All right, welcome everybody. My name is Benjamin Allen. I am a scientific community organizer for the KBase project. I am based out at Oak Ridge National Lab here in East Tennessee, and today we're going to be talking about the KBase platform and introducing everyone to KBase. So, as you can see, KBase is the DOE Systems Biology Knowledge Base. And what is KBase though? It's a community-driven research platform for systems biology. So it's community-driven in the sense that we have, like I said, many tens of thousands of users at this point. Those users publish 150 or so publications a year at this point, doing all sorts of analysis for microbiology, microbial communities, plant microbe interactions, et cetera. And we like to think that we are empowering those researchers to do their work by pulling together data and tools to accelerate their analysis, support collaboration, and help publication. So KBase is a platform that allows us to bring all of those things together and share them with you so that you can do your research. We are supported by the United States Department of Energy, Office of Science, and specifically the Biological and Environmental Research Division. BER, as we call it, supports all sorts of facilities. And you can see with this graphic, there's an integrated approach across these different projects. So some of the projects that we work with closely are the Joint Genome Institute, the Environmental Molecular Sciences Laboratory, the National Microbiome Data Collaborative. And where we sort of fit in this ecosystem is in building a data analysis and visualization platform where all of these groups producing all sorts of kinds of data can integrate their data, their tools, and share them with all of you so that when you're doing your analysis, you can access this data for comparison. I did find new genomes that are related to your project or your research question. So we try to be the platform that serves all of this stuff up in a way that the research community will find particularly valuable. So we're really grateful for BER for supporting us. KBase is about 10 years old at this point. And I've been on the project for 9 years, and I can say that I've seen it grow and develop and really become an exciting resource in systems biology and the biological research community at large. So what I hope to show you today is some of the ways that you can you do the basics of the system and understand sort of the fundamental approaches to the design of the platform and get started on your own work if you're not already a KBase user. Again, these are the kind of things that we do on the platform. We're bringing in data. We allow easy access of open source tools that are very popular and commonly used in bioinformatics workflows. We allow you to build custom analyses and workflows that will help you get what you're trying to do done. We allow you to share the outcomes of that analysis with other collaborators and share those results in your publications as well. And we've got specific features for all of these different priorities with the platform we'll talk about a little bit later on. So getting data into KBase is very easy. For one, we have a drag and drop type interface where that I'll demonstrate later on, where you can just pull files from your hard disk or your computer and put them directly into the web browser interface. They start to upload. So it's very easy to kind of get started with pulling your data on the system. If you have large data sets, and of course, anyone that's been around bioinformatics or systems biology knows that data sets are getting larger and larger. It's getting cheaper and cheaper to sequence more data. So that means that people are getting more ambitious with their projects. We have the Globus platform allows us to, if you have, many terabytes of data, for instance, that, would not be, you would not be able to drag and drop them easily. You can use Globus to pull that into the system as well. But you can see a screenshot of the importer here on the right, and some of those common data types like FASTQ reads or reads from SRA, assemblies, GenBank files, etc. So all of the common genomics data types you can pretty easily get into the platform. There are some new data types that we would like to develop and integrate soon, things like metabolomics or metatranscriptomics data, that, stay tuned for that. But most of the common genomics and metagenomic file formats are integrated into the system already. And this just kind of shows the process here where you can, once you have all of your data imported into the importer, you can just really quickly, bring it into your narratives here. But I'll show that in greater detail later on. We integrate public data resources from, very commonly and essential resources to the biological research community. Of course, RefSeq, the NCBI reference sequence database. We try to, I think, at least once a quarter, pull in all of the genomes, bacterial and archaeal genomes from RefSeq into our public data base. That is important for some of the tools that we have that do, comparison of your genome across all of the genomes in that database. So that's useful because if you tried to install certain tools or do certain workflows on your own computer, you often have to pull all of this data and have enough space and storage and set up references to the data. We try to take care of all of that for you on the platform so that you can get to the actual analysis parts more quickly, and you're not sort of forced to maintain big databases on your own system. We also pull in data from the Joint Genome Institute, in particular the phytosome and microcosm for plant genomes and fungal genomes, respectively. All of the JGI bacterial genomes are imported into RefSeq anyway, so we catch all of those through the RefSeq import. But the idea here is, as there are larger public projects from whether it's the Department of Energy or NIH or other resources of bacterial and plant and basically anything except human data. We do not allow for the analysis of human data on the platform, but basically any other organism, we are trying to identify the kinds of reference databases that are integral to those domains and bring them into the system. But these are some of the key ones that we're starting with. Some of the ones that are coming in the future, for instance, are the JGI viral genomes, as well as data from the National Microbiome Data Collaborative, which is a cross-agency project, as well as the Earth system modeling from groups like ESS Dive. So those are just some examples of where we're bringing in data in the future. And so with those environmental data, for instance, you often have to integrate those experimental and sampling and sequencing data types with metadata about the sampling conditions. So we just last year released the first kind of alpha release of our sample service that we'll keep building on and improving on. But the idea here is to bring together all of these different data types and allow you to manage the metadata associated with your experimental data on our platform. So the idea being that you can link that metadata with your experimental data and as new data from those reference sources comes into the system, or more findings and discoveries are published, you will have these already existing links within your data that you're putting into KBase that you can then extrapolate new findings as more data is integrated into the system. So this is, kind of represents some of the long-term ambitions of the project, where you will have, where, basically as a researcher, you're encouraged to have, highly organized data sets and such that you can um, integrate them into the system. And as the system develops new features or integrates new tools, you'll be able to, constantly reassess the existing data that you have and drive discovery with highly curated data sets. We have a lot of different open source tools. I mean, we've really gone from, like I said, I've been on the project for about 9 years. And, I remember when we had only a dozen or so tools, and now we have over 200 apps. These are all open source tools for doing data analysis. You can see some of the categories here. And we have a software development kit as well that enables developers or bioinformaticians that maybe have custom analytical workflows or tools that they're building to integrate those into KBase so that all of those many tens of thousands of users can access those tools, run the workflows, and publish their findings. So that's a big part of the KBase is giving an incentive for developers to kind of connect with that broader biological research community and put their tools on the system. And here you can see a representation of some of the different flavors of analytical workflows on the system, where if you've tried to do this on your a command line interface, sometimes it can be challenging to create a custom script or workflow that is then able to connect to other types of tools, just because of interoperability of data types often isn't there, or one type of tool generates a certain file that you would like to plug into as another type of analysis, but they can't quite read it. So part of the underlying design of KBase is a data model that encourages the interoperability of data types with these apps, such that you can do a lot of different take a lot of different approaches to analyzing your data, where you're integrating your genomics with your transcriptomics, with metabolic modeling, and just connecting a lot of these different analytical workflows to one another. That's what we hope to see, at least, and we're starting to see our users develop more sophistication with this. Often we get a lot of publications that really do a lot of what's in this top box here with the genomics workflows, but we're starting to see more and more people do combining with genomics, metabolic modeling, or viral analysis, pangenomics, et cetera. I think this is really a good visual representation of really getting the most out of the data that you're generating, because you can really connect it to a lot of other types of analytical workflows. And when you're doing your experimental design, this is always something to keep in mind, that you're setting the limits of how far you can get depending on what types of data that you're generating. So we hope that KBase offers, encourages people to get more diverse and go deeper in the types of data that they're pulling from their samples. So, And all of this is integrated into something that we call narratives. For people that are more computationally savvy, you might have heard of Jupyter notebooks. This is built on a version of the Jupyter notebook. But what you can see here is it's a nice graphical user interface for building out basically a living lab notebook where you have your data, you have apps, and you have all of the analysis sort of in a document where you can basically keep all of your work together. You can write notes, you can even do scripting, and basically this is a persistent document that is shareable. You can share it with other users on the system, you can share it with the world, or you can just run them yourself and try different experiments on the system. So we definitely encourage playing around in the narrative, learning what the different data types and apps are, and I'll show a little bit of that when I get into the live demonstration here. Like I said, we really want this to be a community-driven platform where collaboration is encouraged. So we have some different ways that you can share your data. You can build what are called public narratives. You can also build what are called static narratives that I'll show some examples of. And these are just ways that you can work together, right? Like I shared this link for the questions and answers document in Google Documents. We've all gotten fairly accustomed to using these collaborative platforms where multiple people are working on the same project. So if you have a lab or a group that's working together and you just want access to the same place where you're working on the same data, you can do that in KBase, and that's one of the powerful aspects of the system. We also see educators and professors organizing their classes or their lab groups into what are called organizations, which I'll show as well a little later on. And this just allows you to kind of keep all of your work together, because as we know, whether you're a grad student or postdoc, right, people work on projects and then they move on, and if you're trying to find that data, it's on some old hard drive, things get tricky very quickly. So KBase allows you to keep all of that, those collaborative projects organized on a web platform where you just keep access controlled and persistent. So I'll just keep going on here. So like I said, there are organizations, and in an organization, you can have multiple narratives. You can have different administrators and different levels of permission for controlling access to the organization. So, we see a lot of different kinds of organizations, whether it's for classrooms or for big research projects where they're trying to share all of their data across a series of narratives. So, we encourage you to be creative with this, if and to test what you can and can't do, and let us know if there's things that you would like to do with these organizations. As we develop this system, we are building out something that we call credit engine. But really what this means is that we're trying to encourage folks to get credit for their work, and one of the barriers to people sharing is that there's often a sort of we like to think we own the things that we make, and often science doesn't work well if we're all just kind of hoarding our data. But we want to create pathways for people to share their data, to get credit, and for there to be persistence, and really even bringing that to the whole wide organization level, where you might have a lab that is producing certain types of data that are useful to the broader research community, but they want to let the world know that their lab helped make that data. So building organizations in KBase is one way to do that, where you can enable people to access the data for your group. and create shared resources for the broader community out of your work. The other thing that you can do is include links to your narratives in your publications, and it always makes me happy to see that people are doing this organically now. When they finish their research on the system, they include a link to a public narrative or what's called a static narrative, which, the difference being that a public narrative, you do have to have a KBase account to access the data and to see the work that was done on the system. But a static narrative creates a HTML page that's indexed with search engines, and anyone can look at the data and access the data in it. We're sort of on the 1.0 build of static narratives. So, again, just like with organizations, I really encourage you all to use these features, push them to their limits and let us know what you wish they did or what you think they could do better. That helps us set the priorities for developing the resource in the way that the user community sees to be most helpful to what they're trying to get done. And we're developing partnerships with the publishing groups that we see a lot of activity on. Something like 30 to 40% of those 150 or so publications that come out every year with KBase are microbial resource announcements, where basically people are announcing new genomes that are produced, that they've isolated and assembled and annotated and built a genome with KBase and done taxonomic assessment and so on. That's always very exciting to see because, of course, we, as scientists, we get most of our information from publications. So that helps that kind of feedback loop of community activity when people see that, new genomes are coming out on the system and they're getting published and linked back to on the resource, where you could copy the data and do your own kind of comparison with your own data very quickly. So that's exciting. And this is just an example. I'll pop out of this presentation in a second and show this the static narrative, because it's just an example of one of our user groups kind of organically creating a narrative. And what you'll see here in the bottom corner is that with the static narrative, you can also we can help you generate a DOI for it. So if you are publishing a data set or something that you've accomplished on the system, what we can do is not just create the static narrative, but create a persistent DOI for that static narrative. And that's that can be really useful, not just because it's sort of with things like preprints or pre-publication, you can use static narratives in this kind of way where you're trying to share and then iterate based on the feedback that you get. But it just creates these persistent identifiers that we can link directly to your work on the system. And this is us. So this is our team. We are operating between 4 different national labs, as well as we have university partnerships. We're a small team and an interdisciplinary one. There's computer scientists, DevOps people, programmers, bioinformaticians, biologists, et cetera, that all work together to build KBase as this community resource. So we're really grateful, like I said, for 10 years of support, from going from dozens of users to tens of thousands of users has been really exciting for us to witness. So we appreciate all of you who are, perhaps new to the system and are going to be using it for the first time, as well as those people who've stuck around for many years and watched us develop. So thank you for the support. And you can see the evidence of that with growth of our data, our new users, et cetera, our publication count, and so on. uh, really support, really appreciate all of your support over the years. So, all right, that just links to our project paper, which again, I'll show you here. This is just came out in 2018 in Nature Biotechnology. This describes the vision and idea of the platform. So encourage everyone to take a look at that. There's a link in the Q&A document to this. And also, just want to promote this recent publication of ours from late last year that for people that are interested in microbiome work, this will walk you through doing that analysis. And we had a webinar on this as well in December that you can view on our website, view on our YouTube. Okay, so what I'm going to do now is just, I gave you the presentation version. I'm just going to do a little brief walkthrough through the system to show you some of the different, basic functions and how you do some basic things. And I'll take a look at our Q&A at the end of this, and I'll see if there's anything I can visualize for everybody. Actually, just before I do that, this is just a visual example of the static narrative that I showed in that slide, where you can see there's this persistent URL. I can view, a HTML version of the narrative, check out the data, view even the citations for the tools they used, etc. So this is just a visual representation of the static narrative. All right. So first of all, if you haven't created a KBase account, I should have said this. I think it's in the Q&A doc, but you can just click sign up here. If you have a Google account or an OrcID or a Globus account, it's just very quickly you can authenticate and build a new account with those things. Definitely encourage everyone to link your OrcID to your KBase account. If you have not already, you can do that on the system. Anyway, this is just the KBase landing page. When you go to kbase.us, it's got links to just different ways you can get involved or see what kind of research is coming out on the system. If you want to see news and developments and all of that, you can go to our news page, support on the support page. All that's pretty straightforward. But what I really want to show is the actual narrative interface and the kind of management interfaces here. So when you first log in, this is what you will see is what we kind of call the dashboard area, which just describes the way you can link out to some of the various interfaces on the system. The first thing you'll see is your narratives, your list of narratives that you've created with the system. So you can see I've got quite a few narratives here that I own and manage. These are listed under my narratives. You can also see narratives that are shared with me. So when you are collaborating with people, you're undoubtedly going to share their narratives, share narratives between each other. So I can see all of this list of narratives, click through them. I get a little bit of information about the data that's in those narratives. How much data and of what type is in the narrative. I can even see what information about what tools are run. And this is important because if you're, running projects on, or you might be organizing data and analyzing data from 20, 30, 100 different samples, and you might create 100 different narratives to do each one of those samples. This is just important because it allows you to find what you're looking for very quickly. And you can even search, etc., sort, all that kind of stuff. There's also tutorials. So these tutorials, some of them are associated with protocol publications. Some of them we're working to develop publications around. But if you're looking for a more in-depth explanation of how to do certain common analyses, the tutorials are a really good resource for that. We won't jump into any of these today because we're just kind of trying to intro the platform, but we'll certainly be doing we'll do some webinars on these workflows later this year as well. And of course, you can see recordings of that on the YouTube. We also allow you to look at all of the narratives that have been made public. So, I mean, there's a lot of different stuff. So you can see there's people doing their coursework or sharing narratives for their publications, etc. in here. So this is just a way you can kind of see what other people on the system are doing, and you might find something interesting in there. We are trying to focus over the next couple of years on developing more useful ways to find public data on the system and find public narratives. Looking at features like tagging, if you want to tag a narrative or an organization with a certain theme, or you want to find public narratives that have a certain kind of organism, or some taxonomic reference in them, features like that. So again, this is where we can really use your help. As you're using the platform and you sort of say, oh, I wish I could, click this button and it would show me all of the public narratives with X, Y, or Z. We really appreciate that kind of feedback coming in through our help board. But I just want to create a narrative here so I can show you what a narrative is, beyond just a screenshot. So in the top right corner here, I have this new narrative button, which I'm going to click. And you start to see it spinning up the new narrative here. And this is a blank narrative. So, there's four main parts like I showed in that slide. There's the toolbar up top where we can do things like saving, sharing. I'll just give this a new name. You have to give it a name so that it will save. And so you give it a nice name there. It creates a saved version of this narrative. And now I can do other stuff in here. This welcome cell, and that's introducing the term cell here. As you load data objects or apps, they all go into cells into what's taking up the majority of the screen real estate here, the narrative itself. But I can, for instance, delete this cell if I want to, and then it empties everything out. So I just want to add some data. First, I'm just going to add a piece of public data, but I click this add data slide out here, and sure enough, it slides out a little interface to show me where I can add data from. So I have all of my own data organized across all of my narratives here. If I wanted to find something else that I'm working on, I can also look for certain types. Can also look at the data shared with me. That'll take a while to load, so I'll just keep moving on here. And now I just went to the public tab here where I can see all of the genomes or other data types that are in those public resources that I referenced in the slides. For instance, by default, it starts out with all of the NCBI RefSeq genomes for bacteria and archaea. I'll just type something in here, and I can see I get a filtered list there. That's not filtered enough, so okay, I type in another term and it gives me even greater filtering there. So I find something that matches the taxonomy I'm interested in, or I can even see references to the ID numbers that are used in these other databases, just to know that this is the exact one that I'm looking for. And I just clicked that add button, and that added it to my narrative. So I can now start to play with this piece of data. If I click, underneath the name here, it loads some information about this piece of data. If I click on the name itself, you can see it starts to load it into the narrative. And this is what we call a viewer cell. So you're viewing this piece of data. can see an overview of the data. Since this is a genome, it's it's a list of functions and features and their location and length and where they sit on the assembled contigs. I can I can start to filter by a certain term if I'm looking for a specific feature or protein, and then it will filter down this list of features. Maybe I want to look at this protein and see more information about it. I can see the aliases, I can see the location, the context, and the translation, and as well as the DNA sequence if I want to do any blasting or stuff like that. So this is just showing you very quickly that you can load data, you can start to explore it, you can search for things of interest very quickly. Once you kind of know the mechanics of how to get your data into the system or look at public data. So I'm going to show you now just quickly. I'm going to load some data into the narrative just from my desktop. And sorry, I minimized the cell there. You can expand and minimize cells. Definitely encourage you as you are building narratives to minimize cells if you're not looking at them, because when you are sharing a narrative or you have to load it up, when it has to load every single viewer cell, it slows things down. So that's just a KBase power user tip, is definitely try to collapse your cells if you're not viewing them. So I'm just going to, you may not be able to see it because I'm just sharing one screen here, but I'm going to load a couple of different just from I'm on a Mac, my Finder window. I'm just dragging and dropping a couple things into the narrative here, or into the data importer. And so I have an assembly, a GenBank genome, and it's uploading some short reads here. What you see is after I dragged it in here, it starts to load them. I could terminate that if I wanted to. But once it all loads, it first you're sort of you're uploading it into what we call the staging area, and then you're importing it from the staging area into your narrative. That's important. You might say, well, why can't I just drag it right into the narrative? It's important because you might have multiple narratives that you want to drop the same data into. So you can just you can have multiple narratives where you're then loading data from your staging area. You'll notice here it says all files will be deleted after 90 days. So if you don't load your data into your narrative, it will disappear at some point. That's how we kind of help anybody be able to use the system and do a little fair sharing. But you'll see I just checked all these boxes here, and what that allows me to do next, I checked the boxes after I set the data type. You'll see that it caught that this was a genome and I had to set the other data types. So that's just sometimes there's automatic features like that, but just be careful and check that you're importing something as it really is and not mislabeling what you're trying to import it as, because you may run into errors that way. Just before I click import selected, I'll just highlight there's also ways that you can, like I mentioned, with huge data sets, you're going to want to create a Globus account, and there's instructions about that on our documentation page, how to do that, but you can upload from Globus with this quick link, or if you have a link to a URL or FTP site, you can also quickly import using that. But I've set all these, so I'm just going to import selected, and then what you'll see is a importer app loads in here that has each of the data types. I can set different things about them, like if I want to change the name or set different aspects of, like, what for instance with these reads, can set what kind of sequencing technology they were. That's very important parameter to pay attention to. Or if I have, some assemblers, for instance, require information about standard deviation and mean insert size. So I'm just going to kind of demonstrate it quickly here, but for instance, here with this genome, I can set a scientific name that matches it. Oops, sorry. There we go. I see that this matches the name. That's very helpful in this instance where I already know what the name is. But these scientific names, as you see, they link back to NCBI taxonomy IDs. So that just helps you keep things apples to apples as you're pulling your data on the system. Once I'm happy with everything, I see this run button. It says ready to run. So I'm going to click run. And what you see happen now is that it's running this app. It's running this import job. It's got a batch job in progress. It says I have 3 queued jobs going on, and it shows me information about the what's happening. So some of them are shifting from queued to running. If I wanted to cancel them all, I could click this cancel all button here, but we're not going to do that. But this will just take a couple more minutes. We don't really have to wait around for it, but just to demonstrate, very quickly how you can get your data into the platform. I'm going to jump back to this dashboard viewer here, where you can see I've got these different tabs on the left-hand side here. I just want to go to the jobs tab here just to show you another way to kind of monitor what's going on. So you'll see this has a record of what I have just started there. So it's got links out to those narratives and which app is running. If I click this eye here, this will show me a log of what's going on with the app. And this is just an importer, but any app you can do this with to see exactly the line by line step of what's going on with the importer app. So, and yeah, thanks to Chen in the chat there. Yes, the URL to Dropbox share is a great way to upload data. So yeah, thanks for that tip. Yeah, especially if you already have Dropbox or Google Drive, you can link that to the URL importer. So yeah, anyway, I just wanted to show the jobs tab there. If I go, for instance, previous year, it'll pull out a list of all of the stuff that I've been doing, over the past year. You'll see there's a combination of completed jobs or error jobs. So, if I click this one that had an error, I can go all the way to the bottom and you start to see that red, you had an error. And so this is like, if you want to report something to the help desk, encourage you to go to the log and you sometimes you can troubleshoot it yourself. But if you're creating a ticket, we really appreciate it when you create this this log and drop it into the narrative there. Super. All right. I'm going to go back to this. Now you're starting to see that these pieces of data are populating in the data panel. You see I've got 2 successes, 1 running. Okay, 3 successes. So now all of these different data types were successfully imported into this narrative. You see with the genome here, I have both the genome as well as the assembly. So for instance, if I want to re-annotate that assembly or create a alternate of that genome, I can do that very quickly. I am just going to load this one in here. Or let me actually load the assembly as well, so you can just see some different types of viewers. I can really do this for any of these where I just show you that each data type has its own viewer. For instance, here with these reads, you can see statistics. about the number of bases, or if you want to, look up that information about the standard deviation and things like that, it's all right there. Let's run an app though. That's what we do on KBase is we load data and we run apps. So I am just going to show you how to do something with the genome object here. So if you recall, if I click underneath the name here, I get some different options, to do stuff with the data. So for instance, I can, delete data, I can download data, I can view more information about it. Just kind of pointing these things out so that when you are on your own, you might play around with them and see some of the different things that you can do. I am going to, let's just say, comparative genomics here. So in the apps panel here, I see all these different categories. I can open up different categories, see what's going on as far as the different tools that are labeled that way. But I'm just going to go to comparative genomics here. And I see there's just this big list of tools here. So I'm going to go to this one, insert genome into species tree. It's not very exciting as far as I know exactly what this genome is already as far as what organism it is, but if I had an unknown isolate or something that I wasn't sure what, and I was trying to perform some kind of taxonomic assessment, this is a good tool to use, because what I do is I load my genome into this app, and I can set the public neighbor count. That's the number of branches on the tree that it's going to build based off of that public genome reference database that I was showing. So this is going to build out make a phylogenetic assessment based off of comparison to all of the public genomes on the system. I see these output parameters, so I can set those parameters. And once those little red flags go away, that means I'm ready to run. It will, if I don't have everything set, you cannot run it. But so be sure to set all of the parameters. In other words, if I wanted to add more genomes, so I can actually just, I can add the other one in there as well. So you can see that little plus button. You can add multiple data types there. And if I want to copy any of the public genomes that are generated in this tree into my narrative, I can click this parameter here. So this is just to say you got inputs, parameters, and outputs. Be sure to set them all before you run the app. And sometimes if you get an error, you can revisit those parameters and see what works to try to get a successful outcome. So again, it's in the queue now. I can go back and view it in the jobs browser, etc. What I'll just show really quickly is using a markdown cell. So maybe I want to say something about what I was doing here. I click this little button in the bottom right here, and I get a markdown cell. If I double click it, I can start using markdown language to say, I put the genome into a species tree. There you go. And now I've written some text. If I want to write a lot of text, if I want to link to images or link to other resources or link to other narratives, you can build out all sorts of rich text within the narrative as well. Like I said, trying to build that living lab notebook where you're documenting what you did at different steps. If you're, ran 2 different apps with, different parameters, and you wanted to make sure that whoever came and looked at it later understood what you were doing, really encourage you to, take good lab notes, right? Yeah, just as in the wet lab, in the computational laboratory, you got to take good lab notes throughout the process. So that job is running. It'll probably take another couple minutes here. I can also view the job status directly in the narrative here. Like I can view that log, etc. I didn't really say much about how this app builds the species tree, but the good thing is if you go to the info page here, you can see exactly what this app does. This one uses NCBI conserved orthologous groups to, you know, use a maximum likelihood based method to construct a tree. So this is just, if you're trying to decide what tools you're going to run and in what order, or which one might be the best for your type of data, the best thing to do is read the documentation, and that will help you understand what these tools are. If I go so there's a couple ways you can get to that documentation. I showed clicking the info button here. I'll just also click these three dots, or I can click under the name here. Oh, that actually loads it into the narrative too. But if I click more, what this will do is take me to the full page about this app. And, I can go and see what the publication that references it, or the original publication for the tool is, et cetera. And you'll see this is kind of back in that that dashboard view where it says catalog. In fact, I can go click that or go back to the catalog here. What this is showing is the app catalog. So this is just a different way to view all of the apps on the system. So if you're curious, do we have this app or the other one, this is a good way to look at all of the apps in the library. You can view different versions of apps. If you want to look at beta apps or see apps in development, you can look at those too. We try with anything that's in release, we try to do a lot of testing and authentication that the data that it generates, et cetera, is high quality. But we also encourage people to use the beta apps. If they're in beta, it means you're probably going to get something useful, but we're kind of in the mode of looking for feedback. So check out the beta apps, and in the if you want to test those beta apps, definitely sign up for the beta testers pool. For instance, I think we're just released today a new metagenome assembler, that we would love people to take a look at, called MetaHitmer. But let's see. Some last things, since we're at the end of the hour here, that I just want to show. One is just how to download your data. So, right, you started to generate all of this data. Maybe you want to download some of it. I'll just go to the genome here. I imported it and now I'm downloading it. But if I go to this button here where it says export download data, if I click that, what you see is some different options for standard options, formats for exporting. So I can either download it directly, and this will just download the file to my computer. I can push it back into the staging area. And if you are a computer scientist and you want to understand more about how a KBase organizes its data, this JSON format sort of shows this how the data, all these data types are organized within KBase. So each file that you bring into KBase gets snapped to a data type that is custom to KBase, that enables the interoperability of all of these apps. So a genome produced by any annotator here will be compatible with any other app that takes a genome as input. So that's an important feature, because things don't always work a lot like that if you're working on a file system, or building a custom stack of software. Some other things just to show, I showed the static narratives and shared narratives. I'm just going to go up to this share button here. And if I click manage sharing, I can make I see, first of all, I see that this narrative is private. I can make it public here by clicking this button. There you see, now it is a public narrative. So any if I share this link with any other KBase user, they will be able to open it up on the system and view it themselves. I can share this with specific users, for instance. So you know, if I want to share this with Zach, and I can give him different levels of permission here. You can view only or edit, save, and share. I can just give people different permissions, and you know, if I can give it, I can also take it away by deleting his access to the narrative as well. If I want to share it with a whole organization, I can do that as well. You know, that's a very easy way, right? If you're working with a whole group of researchers to share the narrative with, you know, everybody and not have to tag all of their individual names. And also under this is the manage static narratives. So I'm not going to create one for this. Well, I guess I will. I'll just do it. Why not? So I create a static narrative here, and what it does is it builds out this link to you know, this narrative. This one is not very exciting, but just as a proof of demonstration. If I make, the nice thing about static narratives is that you know, I just created that link, and this is a persistent link. So if I say go back here and start doing all sorts of other stuff, start, you know, loading more apps, more data, that will not be reflected in this static narrative, because this is a snapshot of whatever I was doing in here at a specific time. So that can be useful because you might, you know, for instance, create a version of the static narrative that is shared in a publication, and then maybe you will, you know, 5 years later, a new annotation tool comes out, or new otherwise tool comes out, and you want to rerun that in the narrative that you already had. That can be useful, so you can kind of iterate different versions of your narrative as you're doing different stuff. So okay, we're just 5 minutes over the hour. We won't keep you here too much longer, and appreciate everyone who's sticking around with us. Just a couple more things to show. You know, I had mentioned organizations. I'll just show you the browser here. This shows me all of the orgs that I'm in, but if you just want to see all of the organizations, you can click this filter. And you can see there's links to things that maybe are useful to you, right? Like, for instance, here, you know, Sean is publishing genomes from various NAGs and SAGs. So I could join that organization. You can join that organization if you're interested. And once you click this button, it will send a request to the owner to allow you to you know, participate. You can create your own organization, and that, you know, give it a name, a URL, all of that kind of stuff, provide a description. You can make it hidden or visible, etc. Search, we're rebuilding our search right now, but you know, this allows you to just another panel where you can search any reference data or shared user data. So if you're looking for genome or metabolic model or something like that, you can use the search panel. But like I said, we're kind of reimagining all of our data infrastructure right now, or at least thinking about you know, rebuilding some of the user experience for that. So if you have ideas, again, please drop them into our help board, and that's always appreciated. Feeds here, this, like, for instance, if I sent a you know, request, this would show where you know, where I'm receiving a request to join an organization and such. Again, this is another one where we are looking to reimagine it a little bit, but this is kind of the state of the system right now. Account, you can update your information. You can, you know, if you have multiple accounts, you can link them together. So you have your account information, and if I click on the top right, actually, you'll see a picture of myself and my dog. I can go to my profile, and I can also just, put information about who I am or where I'm at. If you move from an institution and you want to maintain all of your profile accuracy, you can edit that here. Okay, I've given you all a big run through all of the basics of using KBase. Hopefully that was interesting and informative, especially if you're a new user. I am quickly going to just hop back to the questions here and see if there's anything that's been unaddressed that I might, visualize here on the screen. So yeah, because the narrative is built on the Jupyter notebook. Yeah, and I didn't demonstrate this, but if you have, this is something that if you want to use it, I would say reach out to us and maybe describe what you're looking to do, because to show you how to do things like interacting with the data objects and what kind of calls that you need to make for any dependencies to do stuff. It's easier if we can give you some direct support with that, but you can script Python directly into the narrative, but if you're trying to call certain tools and dependencies or libraries and things like that, that's where it can get a little tricky to figure out on your own. So, if you're interested in doing code cell work, again, that's something to maybe request some support on the help board for. Yeah, this is one that we've there's if you go to the one that's called Microbial Genomics Part 1, you'll see information about drafting an isolate genome and publishing it, etc. There is also there's if you type in genome announcement into here, yeah, you should find one that we published a few years ago that kind of walks through that process. But yes, there's information about that on our website. Clear all. See, that's a great feature request. I mean, that's I think these kind of macro functions, as a longtime KBase user, I certainly have my own ideas about some things that we would like to see as far as like functionality in the narrative. For instance, I would love the ability to collapse all of the cells, not necessarily clear them or delete them, but collapse or unfold. Again, we really appreciate when people offer these kinds of suggestions about their user experience. Oh yeah, so I didn't say anything about this, but all of the computational infrastructure behind these apps is built on Department of Energy high performance computers, but split between Argonne as for the production system, as well as at Berkeley Lab for some of the developmental work. And yeah, this appreciate Zach or whoever for putting these tips here, because occasionally when you're running say a big metagenome assembly, you may have you may run into the limits of the system. Part of the reason that KBase is free and open is that it has to be a shared resource for everybody. So there's a limit with running apps, a runtime limit of 7 days. For certain things that are like comparative tools, you can usually expect that they will finish in tens of minutes perhaps, but for computationally intensive stuff like assembly, it's not uncommon to see things go for several days. So if you run something and after a week it failed, it may have not failed as much as it just ran into the runtime limit. We can help you do stuff like break up those jobs and think through strategy for getting it to function on the system. But we are kind of aware, as like I said earlier, as the data types grow and the depth of sequencing grows, depth of coverage, it gets easier to produce those kind of things. We know that the computational intensity increases as well. So, all right. I think I've kept you all long enough. We're about 15 minutes over. I really appreciate everybody sticking around today. We had great attendance, and I hope to see your work on the system. Like I said, my name is Benjamin Allen. I help organize our scientific community here. You can reach out to me at allenbh@ornl.gov if you would like me to come to your institution and do a workshop, or if you have a specific needs like metagenome or microbiome work or isolate work, we'd be happy to chat with you and arrange something where we can get your whole group or your whole department up to speed on using the platform. Just reach out to us at engage@kbase.us and we can chat. But I appreciate, again, everybody attending today. I hope it was informative. And if it was not, you can also create a help ticket for that. No, I just kidding. I appreciate everyone attending today, and thanks. And we look forward to seeing your work on KBase.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.